am Dr. Sandeep Kumar Mishra and I am discussing about various aspects of pharmaceutical microbiology. So, what is microbiology? Microbiology is the branch of science which deals with the study of the microscopic organisms. Microscopic organisms means the organisms which cannot be seen through the naked eyes. So, this branch deals with the study of the microscopic organisms. So, if we discuss about the history of the microbiology history is divided into three main phases. The first is the discovery stage, the second is the transition stage, and the third is the modern microbiology stage. So, in the discovery stage, the initial discoveries which are made in the field of the microbiology were kept. In the transition stage, the discoveries which are in the transition phase between the discovery and the modern phase are discussed, and the modern microbiology includes all the modern recent discoveries in the field of microbiology. So if we talk about the scope of microbiology, the microbiology has spread scopes in various fields. So it involves ecology and environmental application, where the microbiology is used to enhance the environmental protection. Second phase is the food microbiology. The microbiology is also used in the manufacturing and processing of various types of food. Third is the medical and pharmaceutical microbiology. As we know that various antibiotics are obtained from the microorganisms. So this branch deals with the how microbes are useful for in the field of medical and pharmacy. Then vaccines and immunology. So various vaccines are produced using microbiology. And then in the field of agriculture also a lot of discoveries in the field of microbiology are very essential, which are involved in enhancing the agricultural growth. And then in genetic engineering, a lot of microbes are genetically engineered, and these are used for the production of various important products, either of pharmaceutical or medical importance. Then in industry also microbiology is used a lot. Then geochemical application and exomicrobiology. So these are also some of the scopes of microbiology. Then if we talk about the major microbes which are discussed in the microbiology, then these microbes are the bacteria, archaebacteria, which are the ancient bacteria, the fungi, the protozoa, the algae, and the virus. So algae, although is a multicellular organisms, but some forms of algae are also microscopic in nature. So some aspects of microbiology also deals with the study of the algae. Then although microbes grow automatically in the environment, but if we want to culture the microorganisms in the laboratory, then some specific requirements are there for the culture of these microbes. So one of the most important requirement for the culture of these microbes is the culture medium. So what is culture medium? Culture medium is nothing but the food material or the substance which is required for growing microorganisms in vitro, and these are hence called as the culture medium. Hence they are used for the growth of microorganisms outside the body. So if we talk about the classification of the culture media, means how many types of culture media are there, then the culture media can be classified on the basis of the consistency, like solid culture media, semi-solid culture media, and liquid culture media. Then culture media can also be classified on the basis of the composition, like there is synthetic culture media and the non-synthetic culture media. And then culture media can also be classified on the basis of its applications. So on the basis of its applications, the culture media can be basic medium, enriched medium, enrichment medium, or it may be selective medium or differential medium. Differential medium can be used to differentiate different types of microorganisms. Then transport medium. Transport medium is used for transporting the microbial culture from one place to another. And then assay medium. So these are the different types of culture medium. So what are the basic requirements for the formation of the culture medium? So culture medium basically consists of water, which is used as the solvent, the energy source which provides energy to the microorganisms, nutrients which are requirement for the growth of the microorganisms, vitamins and minerals which are important for various important life cycles, life producing mechanisms present in the bacteria or microbes. Buffering agents help to prevent the changes in pH. Then indicator substances which indicates the growth or the selective inhibition of any type of microbe. Selective agents are those agents which are selectively required for the growth of some specific type of microbes. And then finally, the gelling agents. So gelling agents are usually used to enhance the consistency of the media. One of the most common gelling agent which is used is the agar media. So if we talk about the growth of the bacterial curve, or how the bacteria or microorganisms grow, so the growth of the bacteria is basically divided into 4 phases. The first is the lag phase, where just the bacteria are not dividing, their numbers remains constant. Then there is exponential or the growth phase, in which the bacteria rapidly divides and enhance their numbers. Then stationary phase, in this phase the number of bacteria which are producing is equal to the number of bacteria or microorganisms which are dying. And then finally the death phase. So in the death phase, the number of bacteria which are produced is less as compared to the number of bacteria which is dying. So this is overall the growth phase of the growth curve of the bacteria. So if we talk about the different methods used for the isolation of the pure culture, then there is streak plate method. In the streak plate method, usually we use an inoculation loop and dip the inoculation loop in the culture media, and then continuous streaks are made over the agar petri dishes. Then pour plated method. In the pour plate method, the bacterial inoculum is mixed with the culture media, and the culture media is then poured on the petri dishes and allowed to solidify. This is the type of a pour plate method. And then there is a spread plate method. In the spread plate method, a small amount of bacterial culture medium DOP is placed over the culture medium, and then it is spread with the help of the glass rod. Then there is serial dilution method. In the serial dilution method, the bacteria are continuously diluted in the various test tubes, and finally when a very single bacterial cell culture medium is obtained, this can be used for isolation of the pure bacteria. And then single cell isolation methods. Among the single cell isolation methods, there are capillary tube method, or there are micromanipulator method. So if we talk about the maintenance and preservation of pure cultures, then the various strategies which are used is either we can transfer periodically the culture into the fresh media, we can do refrigeration as 4 degrees centigrade. Then there is paraffinization method, or preservation by overlaying cultures with mineral oil, where the paraffin film layer is made over the culture media. Then cryo preservation method. In the cryopreservation method, the bacterial culture is freeze down to the level of minus 190 degrees, 96 degrees centigrade. And then lyophilization method, in which the culture media is then freeze dried. So if we talk about the total and the viable count, how we can identify or do the quantitative measurement of the bacterial growth, then there are 2 different types of methods. There is total count or direct method, and then another is the viable count or the indirect method. In the direct method, there are 4 different methods. First is the breed method, the second is the hemocytometer method, third is the proportional counter method, and then finally 4th is the electronic counter method. And if we want to quantify the viable count of the bacteria, means the amount of the quantity of bacteria which are alive or viable, then there are 2 methods. First is the plate count method, and the second is the membrane filter count method. So this is all about the scope of microbiology, which type of organisms are studied in the field of microbiology, and then how we can identify or obtain the pure culture, and what are the different methods to isolate the pure culture.